In today's video, we are going to study about gram staining. Gram staining method is generally used to classify bacteria into gram-positive and gram-negative bacteria. This method was given by Christian Gram in 1884. Let's see what is the procedure. First of all, we are going to make a thin smear or bacterial smear on a slide, and we are going to heat fix this smear. Okay. After heat fixing of this bacterial smear, we are going to stain this with a alkaline solution of crystal violet. Crystal violet is your primary stain. First stain that we are going to use on bacterial smear is crystal violet. Crystal violet will color it in blue. It will provide color blue to this bacterial smear. Now it will appear blue color. After this, we are going to use iodine solution to fix this primary stain on this bacterial smear. For fixing this crystal violet on this bacterial smear, we are going to use mordant. Mordant is for what? It is for fixing. It will form crystal violet iodine complex. Crystal violet, it is a positively charged, whereas iodine is negatively charged. Both will get combined together. This both will form CVI complex, and this complex will get bounded to bacterial cell membrane. Now this is the cell wall, bacterial cell wall, and here this CVI will get bounded. Okay, it will make an insoluble form. After adding the iodide, it will get bounded, and this complex will get bounded to this cell wall. This is known as, therefore, iodine solution or Ki solution or Lugol solution. These are the names we use generally. So this is used for Lugol solution. Lugol solution. This is used for fixing this primary stain. Okay, it is known as mordant. So in any exam you can also get this question also. What is mordant used in gram staining? The answer is Ki solution, iodine solution, 0.5% iodine solution or Lugol solution. Okay. So after fixing this iodine solution, so now the dye has been fixed on the cell wall, stain has been fixed on the cell wall, So now this fixing of stain on the cell wall is going to appear as blue in color, blue or violet. And we know that the major difference between the gram positive and gram negative bacteria are of cell wall. They have different cell wall composition. So here we are going to see that only. Now this full slide will be washed with water. Okay, after using mordant, we are going to wash with water. After washing with water, we are going to wash it with 95% absolute alcohol or ethanol or acetone. Why we are using this? This will act as decolorizer. It will help to remove the primary stain, to remove the primary stain from this bacterial cell wall. Okay, if the cell wall is lipid soluble, if it is high in lipid content, then if cell wall is high in lipid content, then it will get solubilized in acetone or absolute ethanol or alcohol, whatever you are using. So if we are using this non-polar solution to decolorize the bacterial cell. So if there is a lipid content in cell wall, then it means it is a gram negative bacteria, because gram negative bacteria has a high lipid content, and this lipid will get solubilized or dissolved in this acetone solution. So once the lipid will solubilize in the acetone solution, it can take counter stain also. It means that it has removed its crystal violet iodide complex, the color or a primary stain whatever we have given, it will get removed. Why? Because high lipid containing cell wall has got broken. So now here the counter stain whatever we are using, that is safranine or eosine. Just remember, safranine or eosine is used as a counter stain. It provides pink color. It's going to provide pink color to this bacterial smear. So if this primary stain is removed, then this safranine or a eosine solution will be absorbed over here and it will give pink in color. So those cell which appears pink in color are gram negative, and those cell which appears blue in color, the cell having a high content of peptidoglycan and having a low content of lipid soluble lipid content, will not get solubilized in acetone or absolute ethanol or alcohol. Okay, 95% ethanol or alcohol. It's not going to be decolorized by this discolorizer, and they are going to appear as a blue or a violet color even after using this decolorizer. So now these are the gram positive which appears blue or purple in color because they are having a low lipid content and thicker amount of peptidoglycan in their cell wall. So they are going to absorb that color inside, crystal violet iodine complex inside their cell wall. So they will appear a purple color even after using a safranine. So these are gram positive, and those bacteria which have lost their color, have not retained their color primary stain and took new color safranine or counter staining, on counter staining it appeared as a pink color, they are gram negative. So this is a very small experiment, or you can say small method to classify a bacteria